Good afternoon to everybody, and a warm welcome to our Living Earth Collaborative and the Evolution, Ecology, and Population Biology Fall Seminar Series at Washington University. Today, I have the honor to introducing you all Dr. Iván Jiménez. Dr. Jiménez's long scientific journey began more than 30 years ago at the Los Andes University in Colombia, where he received his bachelor's degree. Then, he received his PhD degree from the University of Missouri, St. Louis, under the supervision of Dr. John Blake. Iván is an Nat Geo explorer and currently is an associate scientist at the Center for Conservation and Sustainable Development in the Missouri Botanical Garden. Iván has an incredible publication record with his work published in Ecology Letters, Global Ecology and Biogeography, and Nature. Iván's current research is focused on testing theories that explain spatial patterns in diversity, structure, and dynamics of geographic ranges of taxa across the Americas, including beautiful places such as the Páramos in the Andes. I want to highlight that Iván is fascinated with the concept of the species, and today he is going to share his work on one of the most charismatic plant species in the Andes, the genus Espeletia. Before to start, two reminders. First, please pose your questions in the chat of the YouTube channel. We will read them to Iván at the end of his talk. Second, if you're part of the LLC community, please join us in the post-seminar discussion with Iván at 5:05. With that, please join me in welcoming Iván. Iván, the audience is all yours.
William, thank you very much for that kind introduction. And thank you to the Living Earth Collaborative for this opportunity to share some of my work. So I think it is widely acknowledged that species delimitation and species taxonomy are fundamental in biology. But I think also it's apparent that not all branches of biology are equally dependent on proper understanding of species limits. I think ecology in particular relies heavily on species taxonomy. And I would like to illustrate this dependence with a couple of examples of studies in which I have been involved. This first example comes from the Madidi project in Bolivia, a long-term collaboration between the Missouri Botanical Garden and the National Herbarium in Bolivia, in La Paz. It has been running uninterrupted since 2001. Currently, this project is led by Sebastián Tello at the Missouri Botanical Garden. So the project focuses on studying the structure, composition, and dynamics of the flora of the northern eastern slopes of the Bolivian Andes. Here is some of the results from the Madrid project, taken from a paper about species rarity, published by Marisabel Loza, who recently finished her PhD in AMSOL working with Bob Rickliffs and myself. This slide shows phylogenetic patterns of rarity among more than 1,000 species, these are all tree species, occurring in 400 tree plots in the Madrid region of Bolivia. Each plot covers about 0.1 hectares. At the center, you see a time-calibrated phylogeny with 134 plant families occurring in the tree plots. The symbols arranged in the three rings around the phylogeny show which families have more or less common and rare species than expected by a null model that randomizes species across the families in the tree. The closest ring to the phylogeny represents local abundance, the next habitat breadth, and the final geographic range size. Most families in the asteroid clade, which is indicated by an arrow, had more rare species than expected by chance, at least in terms of one of the three variables measuring rarity. The point I want to make here is that in this study, as well as in many other ecological studies, species are the basic units to measure rarity in terms of local abundance, habitat breadth, and geographic range size. This is a second example focused on broad-scale spatial variation in species richness, which is a major theme in ecology. This slide references two papers where we focus on estimating the geographic variation in species richness of vascular plants across the Western Neotropics. We also tested different hypotheses proposed to explain this pattern. This kind of research is based on species counts, as so much ecological research, and therefore it is strongly dependent on species taxonomy. And here is another example of research on broad-scale spatial variation in species richness that also depends heavily on species-level taxonomy. In this study, we focus on the diversity anomaly of sister monophyletic plant groups occupying similar moist environments in Eastern Asia and Eastern North America. These groups, on average, have twice as many species in Eastern Asia as in Eastern North America. Again, this kind of research relies heavily on the assumption that species taxonomy is based on a reasonable understanding of species boundaries in nature. This final example is directly relevant to the study system I'll be talking about today. Suppose two plant species replace each other along an elevational gradient so that one species is restricted to the lower elevations and the other one is restricted to higher elevations along the mountain. A common hypothesis about the response of mountain plant species to climate change is upslope displacement of altitudinal ranges, possibly causing so-called mountaintop extinctions, as species occurring at higher elevations run out of suitable habitat. It goes without saying that detection of mountaintop extinctions and generally estimates of the conservation status of a species depend on species limits. So it should be not surprising that ecologists working on these kinds of questions reflect on the nature of a species and on how biologists may go about estimating species limits. Ecologists that do that may identify with Joseph Dalton Hooker, the famous British botanist and explorer that was Darwin's close friend and second director of the Royal Botanical Gardens at Kew. For Hooker, species delimitation was a means to important botanical ends. Hooker was mainly concerned with species delimitation because it was a necessary step for the study of biogeography, which he regarded as a particularly important branch of inquiry. Thus, Hooker saw poor species delimitation and particularly extremely narrow species limits as obstacles to his work on biogeography. I think ecologists may readily understand Hooker. Imagine you're studying interspecific variation in geographic range size or broad-scale patterns of variation in species richness, or biotic responses at the species level to climate change. Now imagine that just as Hooker thought, most work on species delimitation is poor in quality and does not reflect natural patterns. Then your ecological work would be meaningless. So during the remainder of this talk, I would like to first discuss the nature of species from the point of view of an ecologist, highlighting two questions that are generally considered critical to solve empirically current debates about the nature of a species. I will argue that few empirical studies actually address these two questions directly, and that therefore there are important gaps in our understanding about the nature of a species. Second, I will present an empirical study that tries to address these two questions using as a study system the genus Espeletia, a group of plants endemic to high elevations in the northern Andes. Finally, I'll briefly discuss ongoing work and applications of our findings about the nature of Espeletia species. So to begin with broad ideas about the nature of species, it seems fair to say that many biologists think that species are real, meaning they exist independent of human perceptions. Ernst Mayr articulated this view multiple times. Here is an example. The species is a biological phenomenon that cannot be ignored. Every living organism is a member of a species. And this depiction of this is a depiction of the conventional species model that developed from the views of Mayr and other influential evolutionary biologists since the 40s and 50s. According to this model, so-called good species are characterized by distinct and concordant genomic and phenotypic groups. Again, I think it is fair to say that many biologists, by no means all biologists, but many biologists, and in particular, many ecologists, operate under the assumption that this model is a fairly reasonable description of nature. But of course, not everyone agrees. Famously, Darwin seemed to have thought that species were not real, although there's discussion about that, but that's at least one reading of what Darwin wrote. And this quote from the origin seems to leave little doubt about it. But Darwin is far from being alone in taking this position. Here's a quote from Levin: plant species lack reality, cohesion, independence, and simple ecological, evolutionary or ecological roles. Many botanists doubt the existence of plant species. This is from a paper by Risberg. And a more recent quote from Andrew Heap: a measured skepticism about oak species is not uncommon among botanists even today. So you may notice that many of the quotes here are from botanists or describe the ideas of botanists. That is because botanists are famous for doubting the species, that species are real entities in nature. In fact, botanists have been accused of misleading Darwin to believe that species are not real. Moreover, the ideas of botanists about species have been characterized as botanical horror stories. So according to this argument, botanists have been overly impressed by a few examples of organisms that depart from the conventional species model, such as hawthorns, oaks, and rubus, where hybridization or apomixis create blurry species boundaries. According to this argument, these are just a few exceptions, and most plant diversity can be described reasonably well by the conventional species model. This argument, therefore, appeals to the frequency of good species in nature, an issue that can, in principle, be tackled empirically. A number of authors, including those cited in this slide, have presented these two questions as critical to resolve the issue empirically. So first, do individual organisms form sympatric, distinct groups in nature? And second, to what extent taxonomic classification at the species level corresponds to distinct groups of individuals? Next, I'll briefly discuss these two questions in turn, again from the point of view of an ecologist. So the first question emphasizes sympathy because it implies opportunity for any existing groups to merge by hybridization or replace each other via competitive exclusion. So evidence for distinct groups is most convincing when the groups are sympatric. Now, this first question can be empirically addressed using multiple potentially contrasting species criteria, which, as discussed by De Queiroz, are properties acquired during lineage divergence, including, for example, the ability to interbreed, ecological divergence, and reciprocal monophily, among several others. Despite the variety of possible species criteria, there have been enduring interest in the reality of a species in terms of two of them: phenotypic and genome-wide distinctiveness. These two criteria are emphasized, for example, in the conventional species model I discussed earlier. Given these two criteria, one may postulate several working hypotheses about the nature of species. This first one is that there are no phenotypic or genomic groups in the lower left in this table. The second hypothesis is that organisms form concordant phenotypic and genomic groups. This corresponds to the conventional species model, or what many people call good species. In this slide, dots of different colors represent phenotypically distinct individuals, and the dashed lines circle groups according to genome-wide allele frequencies. The third hypothesis is that organisms form distinct groups in terms of genome-wide allele frequencies, but that such groups are not phenotypically distinct. This corresponds to what people often call cryptic species. In this illustration, all points are the same color because there are no distinct phenotypic groups, but the dashed lines show distinct groups of individuals according to genome-wide allele frequencies. Another working hypothesis about the nature of species is that organisms form phenotypic groups that are not distinct in terms of genome-wide allele frequencies. This may correspond to syngamions. A famous example of a syngamion is some oak species. This kind of species may occur when phenotypic distinctiveness is due to differences in few loci that are maintained by natural selection despite frequent hybridization and introgression. In this illustration, the points of different colors show phenotypically distinct groups of individuals, and the single dash circle indicates that there are no genomically distinct groups of individuals in terms of genome-wide allele frequencies. Finally, some species could be characterized by discordant and non-nested sets of distinct phenotypes and genome-wide allele frequencies. In this later case, as well as in the case of cryptic species and syngamions, phenotypes and genome-wide allele frequencies might reflect contrasting evolutionary histories incorporated into a single lineage. While these hypotheses have played a central role in debates about the nature of species, there seem to be few studies that formally test them directly. As I see it, there are at least 3 reasons for this, all related to either failure to obtain data in the first place or problems in the analysis of data. To explain these 3 reasons, I need to go over an explanation of the data required to tackle the question. So phenotypic data and data on genome-wide allele frequencies for a set of sympatric individuals, as represented here by a series of dots. The first step in the analysis is estimating phenotypic groups and separately genomic groups. In this slide, the groups of individuals are represented by boxes around points. The classification of individuals into phenotypic groups is labeled here P, and that into genomic groups is labeled G. Group delimitation should be de novo, and this is really important, meaning starts from scratch. Or put another way, group delimitation should not use a priori information on the assignment of individuals to groups and should be conducted independently for phenotypic and genomic data. The final part in the analysis would be estimating concordance between phenotypic and genomic groups. So now that I explained the data setup and analysis setup, I can explain better why I think there are few studies that answer the question at the top of this slide directly. First, many studies about the nature of species do not actually obtain phenotypic data that allows formal de novo estimation of phenotypic groups. It seems fairly common for species delimitation studies to focus on inferring genetic or genomic groups among individuals thought to represent different species taxa, without formal study of phenotypic data. Another reason why few studies have answered the question at the top of this slide is that even when relevant phenotypic data is collected, the analyses do not actually estimate groups de novo. In the paper cited here, Daniel Cadena, Felipe Zapata, and I discussed the issue and argue that it's quite common in the literature on species delimitation. It should be clear that without de novo estimation of phenotypic groups, it is not possible to assess concordance between phenotypic and genomic groups, and thus it is not possible to address the question at the top of this slide. A final issue has to do with sympathy. As I explained, sympathy is important to test hypotheses about the nature of species in a relatively uncontroversial fashion. Yet systematists working on species limits rarely emphasize intense sampling in sympathy. The current emphasis in systematic biology seems to favor studies designed to sample the whole geographic distribution of monophyletic groups. Due to virtually inescapable logistic limitations, such emphasis results in species delimitation studies that rely on geographically extensive but locally sparse sampling of organisms. So the relative merits of geographically extensive but locally sparse sampling versus localized intense sampling have been discussed in systematic botany at least since the 18th century, famously between Hooker and Asagray. In this photo, both men appeared during an expedition to the Rocky Mountains in 1877. Hooker thought geographically extensive sampling was crucial for species delimitation, which meant that proper species delimitation could only be done from larger varia, like Q, located in the centers of imperial power. On the other hand, Asagray seemed to have thought that the global analysis could be burdened by the difficulty of meaningfully sorting through vast amounts of variation, and that local or regional analysis could yield clearer results. Nowadays, most systematists seem to have sided with Hooker as far as sampling goes, and this is reflected in spatial patterns of botanical collections. Ter Stege developed a model called never the same species twice, referring to the behavior of botanists in a given locality. Again, this model makes the main point here. Studies about species limits are generally based on geographically extensive but locally sparse sampling. And that sampling is just plainly insufficient to decide if there are distinct groups of organisms in sympathy. Now I'll discuss the second question about the nature of the species. To what extent taxonomic classification at the species level corresponds to distinct groups of individual organisms? Risberg and colleagues attempted to address this question in the paper cited at the bottom of this slide and suggested a generally low correspondence. To address this question, it is necessary to distinguish between species and species taxa. In this context, species are understood as objective and discrete groups of individuals in nature that can in principle be detected by looking at criteria such as phenotypic and genomic distinctiveness. In contrast, species taxa are taxonomic divisions at the species level created by taxonomists. So in this slide, I show a classification of individuals into species taxa called T separately from groups of individuals defined by phenotypic or genomic data, P and G respectively. Hopefully making clear that the three classifications are not necessarily the same, and thus it is possible to discuss concordance between species taxa and phenotypic groups, as well as between species taxa and genomic groups. By example, species taxa illustrated here would not correspond to distinct groups of individuals in nature. Such species taxa would be splitting distinct groups of individuals, and that would be the case if one takes as reference genomic or phenotypic groups. In this second example, species taxa would be lumping distinct groups of organisms. Again, in this example, that would be true relative to phenotypic or genomic groups. This kind of comparison between species taxa and phenotypic or genomic groups is also affected by the problems I discussed earlier regarding the estimation of phenotypic or genomic groups. So to summarize this section of the talk, here are once again the two questions that that are generally considered important to resolve the debate about the nature of species. And here is a summary of the three limitations that I think are common in studies about species limits and that prevent these studies from directly informing the debate about the nature of species. Often, there's no collection of quantitative data on phenotypes. Even when that data is collected, there's no formal de novo estimation of phenotypic groups. And often the sampling is not intense enough to ask the key questions in sympathy. So next, I'll present empirical work that tries to address the two questions above the nature that I discussed about the nature of species, while trying to avoid the limitations I also described, using as study system the genus Espeletia. I conducted this work in collaboration with Santiago Madriñán at Universidad de los Andes in Bogotá, Andrés Cortés at the Corporación Colombiana de Investigación Agropecuaria, and Melissa Pineda, that did her master's as part of this project, working with Santiago and myself. We published a first version of this work in bioRxives in case anyone is interested in more details than what I'm going to be able to present today. So we focus on Espeletia, which is a genus of Andean plants that appears to have undergone a rapid recent radiation. There are about 70 Espeletia species taxa currently recognized, but the smallest monophyletic group that includes all Espeletia includes about 140 species taxa from including other genera. So as stated in this quote from the foreword to the most recent monograph of the group, species delimitation in the group may be complicated by hybridization and the recency of the radiation. Here's an estimate of the age of the radiation. At about 2.5 million years, two main clades diverge, and they currently occur in Venezuela and Colombia. But many speciation events seem to have occurred since then, within the last 500,000 years. The thick black line in this slide shows the general distribution of Espeletia, restricted to the Andes of Venezuela, Colombia, and northern Ecuador, and mostly occurring at 3,000 meters of elevation and above, in ecosystems known as páramo. Here's a picture of a páramo in Colombia. These high elevation ecosystems are the primary sources of water for many of the largest cities and agricultural centers in the tropical Andean countries. Plants of the genus Espeletia are dominant in the páramo, as you can see in this picture. Thus, Espeletia plants are thought to be particularly important for the structure and function of these ecosystems. So now I'm going into methods. The study region is the Páramo de Sumapaz, located in the eastern Andes of Colombia, just south of the city of Bogotá. The red line in the image on the left corresponds to a 3,000-meter elevation line and marks the boundary of our study region. The green line corresponds to the limit of Sumapaz National Park. This map shows a digital elevation model of the study region. As you may see, the elevation in the study region ranges between 3,000 and 4,300 meters. We impose a grid of quadrats, each quadrant of size 2 by 2 kilometers, and sample 34 of these quadrats, shown here in black, trying to represent different parts of the páramo. We sample each of the 2 by 2 quadrats using a sampling design that was stratified by elevation, collecting specimens to represent phenotypic variation in each quadrant. We also opportunistically collected specimens across the study region. In total, we collected and measured phenotypic traits in 540 specimens. Additionally, we measure phenotypic traits in 127 specimens cited in the monograph of the group by Cuatro Casas. Here are the 13 phenotypic characters we measured. According to the monograph of Espeletia, 12 of these characters together are enough to distinguish the species known to occur in our study region. To that set of 12 characters, we added one, synflorescence axis perimeter, because based on observations in the field, we thought this character could be useful to distinguish phenotypic groups. The set of characters includes measurements of leaves, measurements of synflorescences, and measurements of capitula and flowers. To estimate phenotypic groups de novo, we use normal mixture models. These models are particularly useful because they can be directly related to theory and species delimitation. The application of these models to species delimitation assumes polygenic inheritance of phenotypic traits and random mating within species. Under those conditions, gene frequencies would be close to Hardy-Weinberg equilibrium, lossy would be near linkage equilibrium, and phenotypic variation among individuals of a single species would tend to be normally distributed. Thus, detection of two or more normal distributions, as shown in this slide, would suggest, especially in sympatric individuals, among sympatric individuals, would suggest the existence of two or more species. So this conclusion would be granted under the assumption that the different normal distributions do not reflect genetic polymorphisms, such as sex-related variation, or ontogenetic variation, or phenotypic plasticity. To measure separation of phenotypic groups, we calculated non-overlapping proportions of the normal distributions representing each group. We call these non-overlapping proportions beta star. More precisely, beta star is the proportion of individuals of each phenotypic group contained within the most inclusive pair of ellipsoidal regions that are 1, non-overlapping, and 2, equal in coverage. Details of how to calculate beta star are in the reference in this slide. But what I would like to emphasize now is that beta star is a metric of phenotypic separation that follows the conventional emphasis that species delimitation studies place on differences in the means of phenotypic groups, regardless of differences between groups in variance or covariance. So the means of the two groups shown here are quite different, quite separate relative to their variation. And therefore, a large proportion of the phenotypic distributions do not overlap, 0.95. If we reduce the differences between the means of these two phenotypic groups, beta star would decrease. And if we go further and eliminate differences between the means of these two groups, beta star would be 0. Yet, there are obvious differences between groups in variance and covariance. So I'll come back to this point several times during this talk. We extracted genomic DNA from 96 individuals out of 540 specimens we collected, and used genotype by sequencing to obtain more than 2,000 SNPs across 77 specimens after filtering for variants with more than 5 reads per locus, 5% minimum allele frequency, and genotypes observed for at least 80% of the specimens. To estimate genomic groups de novo, according to genome-wide allele frequencies, we use ancestry models that, assuming a given number of groups, maximize Hardy-Weinberg equilibrium and minimize linkage disequilibrium within groups. Examples in this slide show admixture graphs illustrating the assignment of individuals to groups, assuming 2, 3, 4, and 4 groups. As you may know, in these admixture graphs, different genomic groups are represented by different colors. Each bar represents an individual, and the y-axis represents, for each individual, the inferred proportion of ancestry from different genomic groups. We adopted an approach developed by Verity and Nichols to estimate the number of groups based on the posterior probability of the models, given the data and a prior of model parameters that is supposed to be uninformative. We implemented this approach with software Maverick. Now, Sympathy, which is key for this study, we define sympathy based on rough estimates of the normal cruising range of individuals, more explicitly defined as the distance between sites of birth and breeding. In Espeletia plants, this distance is likely largely determined by the movement of pollen grains by hummingbirds and bumblebees, because seed dispersal is thought to be fairly limited. Given that bumblebees are thought to be the main pollinators of Espeletia, and that they can regularly fly distances above 1 kilometer, we define sympatric groups as those located no more than 1 kilometer apart. We use the Goodman and Kruskal tau to measure concordance between phenotypic and genomic groups. As I'll point out in a moment, this statistic is particularly useful to test hypotheses about the nature of a species because it measures concordance between two classifications directionally. So in particular, this statistic, JK tau GP, shown in this slide, measures the extent to which genomic groups predict phenotypic groups, as represented by the arrow in the left. The value of this statistic ranges between 0 and 1. Values near 0 indicate that genomic groups do not predict the phenotypic groups, and values near 1 indicate that values near 1 indicate that genomic groups do predict phenotypic groups very well. And there is the metric in the other direction, the JK tau PG, that measures the extent to which phenotypic groups predict genomic groups, as represented by the arrow going from phenotypic to genomic groups. Again, this ranges between 0 and 1. So if the hypothesis of good species, as represented by the conventional species model, is a reasonable description of the data, we should observe that phenotypic groups predict genomic groups well, and also that genomic groups predict phenotypic groups well. So the JK tau values would be high in both directions. In the case of cryptic species, phenotypic groups will not predict genomic groups, but genomic groups should predict phenotypic groups well, because any two specimens taken at random from a single genomic group will belong to a single phenotypic group. Finally, in the case of asyngameon, phenotypic groups will predict genomic groups well, but genomic groups should not predict phenotypic groups. So now consider concordance between species taxa and phenotypic or genomic groups, as shown by the arrows on the left. Note that species taxa are the third set of boxes from the top, labeled T. Again, the classification of individuals according to species taxa may not agree with the classification according to phenotypes or genome-wide allele frequencies. I'll not go into details here, but it is hopefully clear that one can also use the Goodman and Kruskal tau to measure concordance between species taxa and groups defined according to genome-wide allele data and phenotypic data. So next, I'm going to describe how we assign specimens to species taxa. In other words, I'm going to explain how we classify individuals according to species level taxonomy, labeled T in this slide. So we use the species taxa defined in the last monograph of Espeletia by Cuatrecasas. This slide shows 7 species taxa known from our study region. I also included 2 subspecies for one of the species taxa just for illustrative purposes. The 4 species taxa at the top are thought to be endemic to our study region. To assign specimens to species taxa, we use the ranges of phenotypic characters described in the monograph by Cuatrecasas. The color rectangles you see in this slide show the ranges for 2 characters, lamina length and lamina width, for different species taxa known to occur in the study region. I want to emphasize that these ranges are directly derived from the monograph by Cuatrecasas and not from any analysis of the phenotypic data we measure on specimens, on the specimens we collected. In other words, these ranges are drawn based on the descriptions of species taxa presented in the monograph by Cuatrecasas. I would also like to emphasize that even though I'm showing here only the ranges for 2 characters, we use multivariate ranges for all 12 characters that are supposed to be diagnostic according to the monograph of Cuatrecasas. So although the ranges for the species taxa overlap widely in the bivariate space you're seeing here, when the ranges of all 12 characters are considered in multivariate space, the ranges of the species taxa defined by Cuatrecasas do not overlap. So if one of the specimens we collected and measured fell in this point of the phenotypic space, it would be assigned to Espeletia grandiflora, which is the square to which it corresponds. And just to make sure this is clear, it is in principle possible that a specimen we measure falls outside any of the ranges described or defined by Cuatrecasas. So now we go to results, phenotypic groups. These are the results of our approach to de novo estimation of phenotypic groups based on normal mixture models. The model for phenotypic variation with highest empirical support had 6 groups shown here in 3 principal components representing phenotypic variation. The characters that contribute most to the variation in this phenotypic space are shown in the upper part of the slide and include 4 inflorescence traits, 1 flower trait, and 1 leaf trait. In the 2 lower panels, the ellipses represent the equivalent to 1 standard deviation around the mean of each group, and the color points represent specimens assigned to each group according to the legend where groups are labeled M1 through M6. You will notice that some groups are relatively distinct in this space, but also it's very clear that there is extensive overlap between several groups. For example, groups labeled M3 and M5 overlap extensively, although their respective ellipses indicate very different covariance structures. So it is a little bit of a mess towards the center of each of the phenotypic spaces represented in this slide. As a consequence, the phenotypic separation of phenotypic groups showed a wide range of variation. Some pairs of phenotypic groups were clearly separated. By example, phenotypic group M1 was clearly separated from all other phenotypic groups with beta star values equal or higher to 0.9. If you recall the meaning of beta star, these values indicate that a large proportion of the phenotypic distribution of the groups do not overlap. On the other hand, beta star values were rather low for some of the other pairs of phenotypic groups, indicating little phenotypic separation. This was the case for phenotypic groups labeled M3 and M5. Conventionally, this level of phenotypic separation would generally be regarded as insufficient to recognize distinct species. However, it is important to recall that this is a metric driven by differences in means between phenotypic groups, regardless of differences between groups in variance or covariance. I'll come back to this point once again by the end of this talk. All morphological groups were sympatric within 1 kilometer of each other. We conducted spatial analysis to test if phenotypic groups could be explained by variation within a single species induced by spatially structured environmental variation. I'll not show details here of this analysis, but in almost all cases, the analysis suggested that the phenotypic groups could not be interpreted as spatial variation within a single species. Okay, now results about genomic groups. These are the results from our approach to de novo estimation of genomic groups based on ancestry models. This is the model with strongest empirical support as measured by the posterior probability given the data and a presumably uninformative prior for model parameters. As you may see, this model has 3 genomic groups that do not seem to be particularly distinct. These 3 groups are labeled G1, G2, and G3 at the top of the slide. There are many individuals with fairly high proportions of mixed ancestry. About half the specimens assigned to groups 1 and 2 have more than 10% admixture. And fairly high values of admixture are quite common. All 3 genomic groups were sympatric, within 1 kilometer of each other. We also run analysis to test for isolation by distance and see if isolation by distance could explain these groups. Again, I'll not show the details here, but in all cases, genetic distance was always higher between groups than within groups after accounting for geographic distance. Thus, isolation by distance does not seem to explain the 3 genomic groups. Now, concordance between phenotypic and genomic groups. The lower panel in this slide shows the phenotypic group for specimens with genomic data. Not all specimens with genomic data have morphological data, unfortunately. The analysis of concordance between phenotypic and genomic groups based on the Goodman and Kroskol-Tau showed that phenotypic groups predict to some degree the genomic groups, as shown by the value on the left arrow. This result reflects that several phenotypic groups were fully or largely restricted to a single genomic group. In contrast, genomic groups did not predict phenotypic groups, as shown by the value on the right arrow. So overall, this analysis suggests that the nature of Espeletia species resembles a syngamion or discordant and non-nested sets of distinct phenotypes and genome-wide allele frequencies. Finally, what about concordance between taxa and genomic groups and between taxa and phenotypic groups? I'll first illustrate the results of assigning specimens to species taxa. In this slide, the black points indicate specimens that fall into at least one of the species taxa defined by Cuatrecasas in the bivariate space defined by lamina length and lamina width. The gray points show specimens that do not fall within any of the ranges of the species taxa. This slide uses the same conventions, but in addition, the gray dots with black outline show the specimens that fall outside the multivariate ranges defined by the 2 characters shown here and the previous 2 characters, lamina length and lamina width. So only 22 specimens fall within the multivariate ranges of any taxa defined by the 2 characters shown here and the previous 2 characters. Here's the result if you add 2 more characters. Only 17 specimens fall within the multivariate ranges of any taxa defined by the 6 characters. But there are more characters to go. Add these 2 characters and only 10 specimens fall within the multivariate ranges of any taxa defined by the characters shown so far. 2 more characters and only 5 specimens are left, and 2 more characters and only 1 specimen is left within the multivariate ranges of any taxa. So this is remarkable. The specimens cannot actually be assigned to species taxa defined in the last monograph of the group, because most of them fall outside the phenotypic ranges that define those species taxa. I would like to emphasize that this analysis includes 22 specimens cited in the monograph of Cuatrecasas. So conclusions of this part of the talk. Here are the 2 questions I discussed at the beginning of the talk and that seem critical to understand the nature of a species. According to our empirical work, the answer to the first question is that Espeletia in the Páramo de Sumapaz seem to form a syngamion or non-concordant, non-nested sets of phenotypic and genomic groups. As for the second question, I mentioned the possibility that species taxa oversplit natural groups of organisms or lump them, as in this illustration. But what is going is far stranger. And it is not that species taxa are arbitrary divisions of biodiversity. Remarkably, species taxa in Espeletia seem to almost completely miss biodiversity. So talk about botanical horror stories. Espeletia deserves a place here, but not just because it deviates from the conventional species model, but because species taxa are far stranger than many would dare imagine. This finding has many important implications for ecologists and other biologists interested in these plants. Just as an example, consider that the work I presented here today is part of a broader project that includes studies about the responses of Espeletia to climate change and the possibility of mountaintop extinctions. One implication of the results I just presented is that current taxonomy at the species level is not useful to carry out this kind of research. So if I have time, I would like to briefly discuss ongoing work and applications of our findings about the nature of Espeletia species. First, I'll discuss pairs of species with similar phenotypic means, that is low beta star, and different phenotypic variances. I'll focus on 3 points: the biological significance, the detection of these species, and the determination of specimens or specimen identification. After that, I'll briefly mention ongoing experiments that test if phenotypic groups that we detected can be explained by phenotypic plasticity. So as I mentioned, we found phenotypic groups that differ little in terms of multivariate means and thus had low beta star values, but they differ substantially in terms of their variance covariance matrices. The phenotypic groups M3 and M5 are one example. You can see here that these two groups overlap extensively. Their beta star value is 0.39. Yet they differ in terms of variance covariance, as shown by the ellipses that indicate the equivalent of one standard deviation around the means. Conventionally, as I said, this level of overlap would be too high for recognition of separate species, no matter massive differences in variance covariance. I would like to point out 2 reasons why differences in variance covariance are biologically significant in the context of species delimitation, even when multivariate means are identical. First, as we pointed out in the paper cited here, differences in variance covariance of polygenic traits disappear after random mating. So under the assumption used to apply normal mixture models to species delimitation, differences in variance covariance in sympathy constitute evidence of separate species, even when the mean values are the same. Of course, differences in variance covariance may arise by different evolutionary forces, and in some cases, these may reflect differences in ecology of species. So beyond their importance in detection of species, differences in variance covariance may reflect adaptation to spatial variation in the environment, as illustrated by the paper cited here. Thus, even though phenotypic differences in variance covariance have been conventionally ignored in species delimitation, that convention seems at odds with biological theory and empirical work. Perhaps it is time to let that convention go, else studies of species delimitation may not reflect true biological units in nature. But that raises the question of detection. Can species with equal phenotypic means and different variance covariance matrices be detected in phenotypic analysis? In the paper cited below, again, we illustrated this issue with a sample of morphological measurements shown in this histogram, obtained from 2 simulated species with identical mean, but different variance. As shown here, normal mixture models were able to detect the phenotypic distributions of the 2 simulated species in a de novo analysis, with no a priori information on assignment of specimens to simulated species. The work I presented today on Espeletia also illustrates how species with similar phenotypic means but different phenotypic variance covariance matrices can in principle be detected. However, it is important to address the question about detection beyond particular illustrative examples. To do so, Amy Ann, a computer science student at WashU, and I conducted a computer simulation experiment to investigate the performance of normal mixture models in the context of species delimitation. The experiment has a fully cross design with 5 independent variables, including phenotypic separation, as measured by beta star, number of phenotypic dimensions, sample size, distribution of specimens across species, and number of true species. This is a rather large experiment involving the simulation of more than 73,000 species groups. It grew out of a smaller project Amy carried out last summer as a RU student at the Missouri Botanical Garden, funded by the Living Earth Collaborative. Amy decided to expand the experiment, and we're getting pretty interesting results, so stay tuned about that. Finally, assuming that you accept that it makes biological sense to recognize species with equal mean but different phenotypic variances, and that those species can be detected, then there is the question of application in terms of specimen identification to the species level. So suppose the dot shown here along the horizontal morphological axis represents a measurement of a particular specimen, say specimen 1. The question is which of the two species the specimen belongs to, species A or species B. The straightforward way to apply normal mixture models to specimen determination is using maximum likelihood. So here specimen 1 would be assigned to the species A. And a second specimen labeled here specimen 2 would be assigned to species B, represented by the dashed line. You may be thinking that using maximum likelihood in this way may lead to a lot of identification errors, particularly for specimens located near the mean values of both species. And that is true when the number of phenotypic dimensions is low, as in this case, it's just one dimension. However, as the number of phenotypic dimensions increases, the error rate decreases. More so if the species differ not just in the size of the variance covariance matrices, but also in the shape of the variance covariance matrices. Very good rates of correct specimen identification can be reached with 10 phenotypic dimensions, sometimes less. I'll not get into this now, but this is partly related to a property of normal distributions described by the Gaussian annulus theorem. We're applying these ideas about specimen identification in a project involving these 5 co-investigators, Erika Benavides and Moises Penagos, shown at the top, are campesinos from the Páramo de Sumapaz interested in environmental issues. Juliet Avila, Marco Pardo, and Luz Dari Rodriguez work for the National Park Service in Colombia, and monitoring populations of Espeletia at the species level is part of their mandate. But as I have shown, the current taxonomy does not seem particularly useful to do so. So as a first step, we are developing a guide to the Espeletia species from Sumapaz. The guide is still in construction, but it will include taxonomy according to the monograph of Cuatrecasas, even though we think that's not particularly useful. But it also will include a probabilistic and interactive key that applies the principles of species identification by maximum likelihood that I just discussed. Now the guide has an R code that illustrates sort of the principles of how one could identify specimens using maximum likelihood. Another major component of this project involves testing whether the phenotypic groups we found can be explained by phenotypic plasticity with similar phenotypic plasticity within a species. As I mentioned earlier, we conducted spatial analysis to test if phenotypic groups could be explained by variation within a single species. In most cases, the analysis suggested that no, they cannot. However, one of these analyses suggested that 2 phenotypic groups labeled here M3 and M5, this slide, could be interpreted as either phenotypic variation induced by elevation or as 2 species occurring at different elevations. This can be seen in this slide where the y-axis is phenotype and the x-axis is elevation. So we're conducting, to test these 2 hypotheses, we're conducting common garden experiments. And here's a pilot of the common garden experiment that started in April last year in the farm of Don Moises Penagos, one of the local co-investigators in this project. I should say Espeletia plants have a reputation for being difficult to grow in greenhouses, presumably dying soon after germination. But Moises, shown in this picture, and Erika Benavides, the other local co-investigator, have been successful at maintaining these plants for well over a year. Finally, I would like to thank these 5 students from the biology department of the Universidad Nacional de Colombia. They contributed substantially to fieldwork, and 4 of them did their undergraduate thesis as part of this project, co-advised by Dolores Armenteras and Maria Genis Bonilla. We were supported by many local people during fieldwork, and I was helped by multiple discussions with Peter Stevens, Kate Wasselkoff, Amy Ann, and Jill Preston. And as I mentioned earlier, this work was part of a wider project about the responses of Espeletia to global change that was originally designed with Cesar Marin and Carlos Lora. The Jardín Botánico and the National Park Service in Colombia have supported the project since it started in 2017. And I thank multiple sources of funding. And thank you very much for listening to me.
Many thanks, Ivan. That was really intense and fascinating talk and really complicated too. So while we're having the questions coming, there's one question of conservation. So it's so hard to limit the species or the good species. So if we apply this approach, for example, to these keystone species or ecosystem engineering species, should we like redefine those to do conservation?
You mean Espeletia as key species, or you mean more generally?
In general, I think it's like more in general.
I mean, I think it depends on the case, right? There might be species limits in some keystone species or important species, maybe okay. I think in the case of Espeletia, what I think this kind of approach would foster is actually paying attention to species limits and doing observations with measurements of phenotypes. So that even if you don't believe the results of this particular study, you have the phenotypic data and can actually eventually come up to use that data to come up with other delimitation if you think this is not useful.
Great, thank you. We actually have a question here from the chat. So Brock Mashburn asks, what kind of genomic data was used at the admixture analysis, and do you think this affects the results? And then secondly, would the only appropriate genomic data for these analyses be whole genome sequences?
Well, the data we use is a bunch of SNPs, like 2,000 SNPs. And I mean, that seems good, but a lot of studies have way more than that. We don't know where those SNPs are, of course. We cannot estimate linkage, for instance. That would be nice for an admixture analysis. So I think, yeah, I mean, more data can change this, of course. Now, genome-wide, well, I defined as one of the criteria, species criteria, genome-wide allele frequencies. Other criteria may be used, but I think that's a common one, or it's interesting from the perspective of the development of species concepts, because it has to do with the unity of the genotype and how that was challenged by the genic view of speciation in early 2000 by Wu, and I guess by botanists way before that. So I think I'm not arguing it's good or bad. I'm arguing that it's an important species criterion, genome-wide allele frequencies.
Thanks, Ivan. So the time is a constraint now. And so we want to thank you again for this fantastic seminar. And let's reconvene at 5:05 to chat with Dr. Jimenez. All the ALC fellows, please do it. And Ivan, you have a lot of questions in the chat, so we will pause those, and then you can reply them by on the chat or by sending emails to them too. So thanks again, and please, all the audience, stay tuned for the next ALC seminar series.
Thank you very much.